Yeah, I'm going to talk to you about mapping some mycorrhiza from the Harrison lab. And thanks to Maria for providing a nice background onto the symbiosis. If we zoom in here, this is the plant cortical cell where we have fungus coming into that periarbuscular space, and it's able to provide the plant with such things as phosphorus, nitrogen in return for the lipids and sugars that it needs to live. And you can see this here labeled with phosphate coming down the hyphae, coming into this arbuscule, and then providing it to the plant. So what does this look like on a root scale? These are nice pictures from our undergrad Amita that she took. Here you can see the fungal labeling inside the plant root. And one of our new colonization promoters that we found through this work, you can see the actual arbuscule standing on those inner cortical cells surrounding that epidermis. So we know what's happening on the plant in on a whole root scale as far as the total RNA expression, but we're not entirely sure what's going on that single cell scale, what's going on in the ones that contain the arbuscules, what's happening when the arbuscule progresses, and then what's going on in the other tissue around. And so to get through this questions, I undertook the project of undergoing spatial transcriptomics in plant roots. And what this does is it basically takes some sections of your plant roots. It's going to permeabilize, drop the RNA down onto these capture spots. Each of these capture spots has a bunch of these poly DTs that'll capture that mRNA, and they'll attach this spatial barcode onto it so that when you sequence it, you can figure out where exactly on your map those mRNA are coming from. And so with this, I undertook this experiment. I used a colonization marker to find those little plant roots with a microscope, cut them up, and basically cryopreserve them to make sure that RNA stays nice and fresh, section it onto slides, and then drop that RNA. Here you can see a cDNA footprint with a conjugated fluorophore. But once we get this, we can then sequence it and then see what's underneath, which is going to be the interesting stuff. So here on the left, you can see here are some non-colonized roots and colonized roots. And what you see here is just the normalized gene counts of some fungal reads. And so you can see fungal expression is much higher. Obviously, there's fungal reads present in the colonized places. But we can also look at this on a single gene level. Here's one of our favorite genes, PT4. It's a symbiotic phosphate transporter that occurs in these arbuscules. And you can see individual spots here containing those cells that have that expression from that symbiotic phosphate transporter. And so we can see exactly where those active arbuscules are in the plant. We can look at the bulk differences, obviously, using this with roots and compare it to previous RNA-seq studies that previously used bulk colonized versus non-colonized gene expression studies. And we see strong differences that sort of recapitulate those findings, recapitulate the findings of these also arbuscular mycorrhizal conserved genes that Maria was talking about through evolution have been conserved over the 400 million years. Those are present over here and upregulated during colonization. And importantly, we can find evidence of, so this is the progression of an arbuscule starting with the formation of it, development, and then this fine branch formation, which has the actual nutrient exchange occurring. And this is, like all things, temporary. It degrades over time, in this case, on like a week level basis. And so we have a couple markers that we know that can mark these degenerating arbuscules. And all of these genes are present in our study, and hopefully we can segregate them out into the different places in the root and see what's expressed along with these marker genes that we previously have. And so I'll talk about using this phosphate transporter to show where these cells that have nutrient exchange occurring, as well as a senescence marker. And we can just look at a couple examples of what's going on in these cells happening in these two distinct arbuscular cortical phases. And so if we go into this, this is in where nutrient exchange is occurring. We can see such things like German-like proteins coming through, some of this has been found, a similar protein has been found in rhizobial symbiosis that involves membrane protein interactions. So interacting directly with that bacteria's protein membrane. In this case, we're doing fungi, obviously, but this is involved in some sort of plant defense. Stabilization of the periovascular membrane, we find a remorin that's thought to help create and preserve this really convoluted structure that's useful for all that nutrient exchange, that surface area. And we also have signaling pathways that we find are both shared between rhizobium and arbuscular mycorrhizal fungi. We have some genes here, a couple ones that are show this activation of this pathway, as well as some other transcription factor genes that are not yet described in this process. So all that is important to make sure that these things happen, mostly this nutrient exchange. We've talked about the phosphate transport. That's obviously happening in these genes that we've selected for phosphate transport. But we're also seeing ammonium transporters coming through in those cells, as well as ones involved in microbial defense. And by microbial defense, these are genes that are related to other genes that are involved in kicking out pathogens, but these are seen in a symbiotic context. So maybe there's some sort of additional role they're playing in this, maybe recognition or something, or waiting for it to turn into a degenerated arbuscule. We also see some of these undescribed kinases, some of which we've been working on making sure that they're established in these arbuscular structures. And so there's a whole large list of these hypothetical genes that we can go down and try and understand what's going on. On the senescence side, so the degenerating arbuscule side, it's been known that chitinases and proteases and lipases have played a role in trying to degrade a lot of that structure and creating these sort of monkey fists that you see at the end. And we find the exact same thing when we use our study to look specifically at those degenerative cells. We see chitinases, proteases, right-binding related proteins, some lipases. And then we also see a good overlap with one of our previous studies in the lab, which used a PT4 mutant, a phosphate mutant, which created a lot of degenerate arbuscules in the plant. And so you're seeing a lot of these that we saw on the whole root level be recapitulated on this individual cell level. So we know that this is happening inside these cells. We also have a bunch of other genes that we can go look at and try and infer what's going on in that plant. Well, with this, we can also look at the fungal genes that are associated with these different stages. And one important one is to deal with how rhizophagus, or the arbuscular mycorrhizal fungus, utilizes the fatty acids that it's receiving, or the lipids that it's receiving from its host. And in this case, there's a desaturase that is highly upregulated in the arbuscule that is making sure that these arbuscular mycorrhizal fungi can make these long-chain fatty acids that it's getting, the precursors it's getting from the plant. In the senescent cells, we see a little bit of a different story. We see a little more immune sort of evasion sort of happening, at least for the ones that are annotated. There's a lot of unannotated fungal genes, as you might expect. We have something like chitin deacetylase, which is one way for pathogens or symbionts to avoid the immune system of their host, as well as this isoflavone reductase. And isoflavonoids are one way that plants can use to basically locate and either stop the growth or make sure they know what's going on where that fungus is active inside the host. And so these are two ways that the fungus could possibly be trying to deal with this plant degenerative program that's occurring. So we've looked at how we can use plant markers to isolate these different parts, but we can also use the microscopy itself, in this case, to identify those spots that contain mature arbuscules or collapsed arbuscules and see what's happening underneath. And in this way, we can also add an unsupervised way of looking at it by looking at these gene correlation networks that look at what's upregulated in some of these spots. So here we see upregulation a module that's upregulated in these arbuscular regions. And we can we can get the network, the gene network that comes out of there that contains both plant and fungal genes and shows how these genes are sort of connected as far as which ones are more closely are more closely mimicking each other's patterns, spot to spot. And we can see, so one of these is the desaturase, and a bunch of other of these are peptides. But they're, going up and down with some of these important plant genes. So arbuscules aren't the only fungal structures that are occurring inside colonized plant roots. We also have fungal structures like vesicles and spores that the fungi makes for either lipid storage or propagation. And so we can use this, and there's less of them, so it's a little bit more difficult to draw statistical power from it. But we can find modules that also are upregulated in those areas. If we want to get more statistical power, what we can do is annotate using our single nuclei data, which we used. And this is useful because these vesicles and spores, they have many more nuclei inside of them. And so when you chop this up and sequenced it, then you can get the statistical power much higher, and you can actually get separation between some of these structures. And so we can map these back onto the plant maps that we have, and we can see actually some of these things that look morphologically extremely similar, they're mapping onto very different spaces in 2D. And everything here, the closer it is together, the more similar its transcriptome looks. And so if we just take one of these, for example, we can see that this module of this cluster that's upregulated in this cluster has very high levels of histone activity, as well as some centromeres and chromosome segregation, things that are involved in mitosis, DNA proliferation, replication, things like that, which led us down into maybe this is actually different than what's going on in here, or maybe there's a continuum between these three different stages of the vesicle. I tried looking for DNA replication in fungi in the plant. I was able to get some nice replication in the actual plant cells themselves. No information yet on whether vesicles or anything of these structures are getting any replicative DNA in there. So this is still in progress, but I'm interested in going further down this road. So why are we interested in there? Why do we care about this? Because one reason is because with these fertilizers, there's a lot of nutrient pollution that occurs when you have storm runoff like from today. You can turn a whole Great Lake green, for example. And so when we're using AMF and even the microbes in the soil, we can hopefully breed crops to better take advantage of this symbiosis. And so with that, I hope I've kept it to under 15 minutes.
Awesome. Any questions for Trevor? Yes.
Yeah,
a fascinating talk. I was wondering, because you guys are assuming a lot of cool stuff in terms of AM symbiosis happening in the cortex. I was wondering, do you also see changes in other cell layers that are providing more context and accommodation for the arbuscular mycorrhizal symbiosis?
Yes, so there are changes that we can observe because we have both the single nuclei maps too. And you can see the changes that are occurring in the stele and the phloem and xylem, for example. We have lists of genes that are changing in the endodermis. And then it's a little difficult teasing the epidermis and the cortex apart, but there we have some modules that are clearly being upregulated in the epidermis of colonized cells, which are interesting. So yeah, Andrew.
You mentioned some peptides coming from the fungus to the plants. Any clue what's going on there?
Yeah, it's hard. I'm not entirely sure. A lot of this, most have been sort of annotated on a basic level. So I've been having to go through sort of gene by gene. Really, a lot of these are fairly short proteins, at least the ones that are here. I'm not entirely sure what their structure is. A lot of it seems like what's the word? Disorganized?
Disordered, yeah.
Disordered, yeah. So
and are they co-expressed with other groups of genes that might clue you in as to function? You have the network there.
Yeah, so some of them are involved, a lot of are involved in like RNA binding and splicing and maybe a little bit. Yeah, I think it's RNA binding and RNA splicing actually that popped up.
Maybe we have another. Apologies if I missed this, but what were you hoping to find with the fungal DNA replication in the neurovascular vesicles?
So I'm looking to see if, so there were 3 groups of those vesicles that I was able to disentangle in the single nuclei data, but morphologically they all look the same in the plant. And so I'm trying to see if one of those groups is perhaps more involved in DNA replication and repair than the others. And if that's the case, then we would, if we're trying to get those DNA to be labeled, we would see maybe just 1/3 of those structures being labeled with that. Or maybe we would see a gradient if in case it's actually progressing over time.
Oh, okay, that makes sense.
Yeah. Yeah.
I think you mentioned this, but can you remind us when you lay the sections down, like how thick are the sections? How many cells thick are they?
How many cells thick? So these are 14, actually 18 micron sections. They can vary depending on the cell type. So it can be between maybe 1 to 2 cells thick, but in some areas it could be even 4 cells thick, maybe. Yeah.
And so I'm wondering, it seems like you have really nice boundaries of these sections, and I'm wondering if you might be able to tease out some subcellular information from that.
I think it'd be a little difficult at this resolution for the spatial stuff. It's still at maybe like the 2 to 3 cell level, I think. But with the single nuclei, there's ways, computational ways that you can use to get at a smaller resolution. So hopefully going forward, we're able to get more at that.
Are there any other questions for Trevor? Let's give him another round of applause.